Hello guys, I'm Zeri from Ace Academy again, and today I'm gonna solve 9700 October November 2022 variant 12. So let's begin. Number 1. The diagram shows a stage micrometer with divisions 0.1 millimeters apart viewed through an eyepiece containing a graticule. The area of the field of view of the microscope can be calculated using the formula area equals to pi r square. A student calculated the area of the field of view of the microscope using the information provided and a value of pi for 3.142, which answer has been rounded correctly to an appropriate number of significant figures. So this is a typical graphical question. Firstly, we have to identify which scale is which. So the above scale over here, this is the stage micrometer. Stage micrometer, and in every microscope there is a scale that has 0 to 100, and this is called the eyepiece graticule. Okay, that's great. So the eyepiece graticule does not have any units. It depends on the magnification. If we magnify more, then the eyepiece graticule unit becomes lower, and so on and so on. So the question says that the stage micrometer has division 0.1 millimeters apart. So this entire thing is 0.10 millimeters apart. And this refers to 15 minus 10, which is 40 gratical units. So 40 eyepiece gratical units equals to 0.1 millimeters. 1 eyepiece gratical unit equals to 0.1 by 40 millimeters. Therefore, this is 2.5 into 10 to the power minus 3 millimeters. Then, the radius of the entire field of view is this, 0 to 50. So, radius equals to 50 into eyepiece gratical unit, which is 2.5 into 10 to the power minus 3 millimeters. And then the field of view will be pi 50 into 2.5 into 10 to the power minus 3 whole square, which gives us an answer as 0.49 0.04909 mm square. So this is an option over here, but there is also another thing that is working here. Which answer has been correctly rounded to an appropriate number of significant figures. So here, the number of significant figures is 5, which is a bit too much. And in microscopes, we usually use micrometer more than millimeter. More than millimeter. Therefore, this will not be correct. Instead of this, we have to convert this to micrometers. So 10 to the power 6, it's 4.91 into 10 to the power 4 micrometer square, because 1 millimeter equals to 1000 micrometer. 1 millimeter square equals to 1000 micrometer square. 1000 square micrometer square. So the overall answer over here is D. Now, question number 2. Where are cisternae found in a cell? Well, cisternae are flattened fluid-filled sacs. Flattened fluid-filled sacs. So, cisternae, endoplasmic reticulum has cisternae, Golgi body has cisternae. However, mitochondria doesn't have cistern. So what the answer is A option of question number two. Okay, now let's move on to question number three. Which row matches the functions to the cell structure? Vacuum stores water, sugar, and pigment. This is correct. Some other functions of vacuoles are stores cell sap and provides hydrostatic support. We will see if these are present in the other options as well. And by hydrostatic support, I mean turgidity. It helps with keeping things turgid. So this is correct. Nucleolus. Nucleolus manufactures messenger RNA. No, it's incorrect. It's actually rRNA. Then ribosomes, protein synthesis. Yes, this is correct. And lysosomes, involved in cell recognition. No, it is not correct because lysosomes contain hydrolytic enzymes. Okay, let's look at B. Exports proteins. No, it doesn't. Nucleolus manufactures rRNA. Correct. Ribosomes, lipid synthesis. No. Lysosomes involved in cell recognition. No. Number C. Stores water, sugar, and pigment. Correct. Manufactures rRNA. Correct. Protein synthesis. Correct. And lysosomes contains hydrolytic enzymes. Correct. So C can be the correct answer. Let's look at D. Vacuole exports protein. Again, this is incorrect. Messenger RNA incorrect. Lipid synthesis is incorrect. Lysosome contains enzymes. This is correct. So C is the only correct answer for number 3. Number 4. Which structures are found in prokaryotes and eukaryotes? Cell membrane and nucleus? No. Prokaryotes do not have a nucleus. Their DNA is naked in the cytoplasm and it is not associated with any proteins and it is circular. So option A is wrong. Cell membrane and ribosomes. This is correct. Prokaryotes have 70F ribosomes while eukaryotes have 80F ribosomes. Nucleolus, nucleus and mitochondria. No, prokaryotes do not have mitochondria. Mitochondria and ribosomes. No, prokaryotes do not have mitochondria. So B is the correct answer. Number 5. Which molecules are present in all viruses? Thymine, not really, because uracil can be present if it's RNA. Adenine, yes, it will always be present, and cytosine is always present. So number 2 and 3, which is D. Number 6. The table shows the observations recorded from tests for biological molecules on 4 samples A, B, C, D. Benedict solution blue. Its initial color was blue as well, so no reducing sugar. So option A and B, both of them have no reducing sugar since their color remains blue. Now, biuret reagent. It's blue and its initial color was blue as well. So no protein present in A, while protein present in B because the color changes from blue to purple. Iodine. The color changes from orange to blue black, so starch is definitely present and starch absent in these two. Again, no protein in these two, starch present in B, and reducing sugar present in C and D. So overall, A contains starch only. This is correct. B contains reducing sugar only. No, B doesn't contain any reducing sugar at all. C contains reducing sugar and protein. No, it doesn't contain protein. And D contains starch and protein. Yes, it does contain starch. However, it doesn't contain protein. So A is the only correct option over here. Number 7. The diagram shows some biological molecules. Which biological molecule forms a polymer with a structural role in plants? Well, by structural role, we mean cellulose. And cellulose is made up of monomers which are beta glucose. So, we have to identify the beta glucose over here. This is definitely not beta glucose, this is alpha glucose because the hydroxyl group in carbon number 1 is below the plane. In beta glucose, it's supposed to be above the plane. So it's definitely alpha glucose. And this is just a random alcohol molecule, so this is incorrect as well. And this is something that you're not required to know in the syllabus. And D, let's look at D. Carbon number 1, hydrogen above the plane, below the plane, above the plane, below the plane, and this. So this is indeed beta glucose. So D is the correct option over here for question number 7. Question number 8. What is a disaccharide and a monosaccharide and a reducing sugar? Well, both of them, fructose and glucose, these are monosaccharides. So these are automatically wrong. Both maltose and sucrose are disaccharides. However, sucrose is a non-reducing sugar. That leaves us with only maltose is the answer because maltose is both a disaccharide and a reducing sugar. So, question number 9. The diagram shows bonding in part of a polysaccharide. Which type of glycosidic bond is shown at position X? So there is no flipping of the bond, like no flipping like this. So this is definitely not cellulose, and this is definitely not beta 1,6. So beta 1,6 can be crossed out, and this is carbon number 4, this is carbon number 1. So this is basically alpha 1,6 glucose. And over here, this is carbon number 5, 6, and then this is carbon number 1. This is alpha 1,6 linkage. So X, the answer for question number 9 is that it's alpha 1,6 linkage. So for 9, B is the correct option. Number 10. Which statements about triglycerides and phosphates are correct? Fatty acids in a triglyceride may be saturated or unsaturated, but in a phospholipid they are always saturated. Incorrect, because phospholipids can be saturated as well. Unsaturated as well. They might have double bonds in their fatty acids too. So one is wrong. Triglycerides and phospholipids both have a hydrophobic region, which is the fatty acid region. So this is correct. And then triglycerides are non-polar molecules, while phospholipids are polar molecules. Yes, phospholipids are polar molecules due to the presence of the phosphate group and the choline group. And triglycerides are non-polar due to the presence of glycerol and fatty acids, both of which are non-polar. So 2 and 3 are correct, which gives us with C as the correct option. Question number 11. Silk moth caterpillars have been genetically modified to produce a mixture of their own silk and the much stronger spider silk of the golden orb web spider. The spider silk polypeptide chain, so note how it's singular, so one polypeptide, has many repeated sequences of two amino acids, glycine and alanine, arranged in crystalline structure. What correctly describes the structure of spider silk? So when we have these structures, these are called beta pleated sheets. Beta repeated sheets. So this spider polypeptide has a lot of this beta repeated sheets. And when we have these are called alpha helices. And both of them are secondary structure. So, what correctly describes the structure of spider silk? Primary protein structure, yes, there is. This can be called as a primary protein structure because there is talk there because this MCQ talked about many repeated sequences of two amino acids, glycine and alanine. Then it primary protein sequences with regions of alpha helices. No, these are beta repeated sheets, so this is incorrect. Secondary structure, this is true, with regions of beta repeated this is true as well. So B can be correct. Tertiary protein structure with regions of alpha helices. No, because there are no evidence of alpha helices in this polypeptide. Then quaternary protein structure with regions of beta helices. As I told already earlier, there is just one polypeptide, and there cannot be a quaternary structure with just one polypeptide. Thus, D is incorrect as well, which leaves us with only B as the correct option. Number 12. Which property of the tertiary structure of a globular protein enables it to catalyze a metabolic reaction? Does hydrophobic amino acid R groups on the outside? No. Hydrophilic groups are on the outside, while hydrophobic groups are on the inside. So this is opposite. B. It will be denatured by high temperatures. Well, no, not really. This isn't a property that enables it to carry out a metabolic reaction, right? So this is incorrect. The R groups of some amino acids form bonds with the substrate. This is true. For example, ionic bonds can be formed, hydrophobic interactions can be formed, and then hydrogen bonds can be formed, and this helps to stabilize the substrate with the globular protein. So C is correct. The three-dimensional structure, the shape depends on hydrogen bonding. Well, this is correct, but how does this enable it to catalyze a metabolic reaction? It is not explaining this. Okay, so D is incorrect. Thus, let's move on to question number 13. A student investigated the effect of a substrate concentration on the activity of an enzyme. The graph shows the results of this investigation. An increase in which factors could lead to a change in the activity of the enzyme at point A on the graph? But number one, pH. Well, over here, substrate concentration is limiting. Substrate concentration limiting. Okay, we inferred that. This is this could lead to a change in the activity of the enzyme, yes. And pH could also lead to a change because increasing or decreasing pH might even denature the enzyme, rendering it unable to catalyze the reaction. And temperature too, because increasing the temperature can denature the enzyme as well. So all three are correct, which leaves us with A as the correct option. Question number 14. The Michaelis-Menten constant Km is the substrate concentration at which an enzyme works at half its maximum rate. What is correct when the Km value is low? When the Km value is low, it means that enzyme more efficient. High affinity for the substrate, meaning it can bind to the substrate more easily. And it also means that less substrate required for Vmax. Vmax is the maximum rate of reaction, so faster, shorter time to reach Vmax. So basically, graphically, it can be shown as something like this. So this is Vmax over here, and this is substrate concentration. An enzyme with lower affinity would take a more substrate to reach the same Vmax, while an enzyme with higher affinity would take less substrate to reach the same Vmax. High affinity, money, or low Km value, low Km, high Km. Okay, so let's look at the options. What is correct when the Km value is low? The enzyme has a low affinity for the substrate, and the quicker the reaction will proceed to its maximum rate. No, it's actually high affinity. So this is incorrect. The enzyme has a low affinity for the substrate. Again, this is incorrect because it's high affinity. And the slower the reaction, no, it's quicker. So this is completely wrong. The enzyme has a high affinity for the structure. This is correct. And the quicker the reaction will proceed to its maximum rate. This is correct as well. Coordinate C can be the correct answer. And then the enzyme has a high affinity for the structure. And the slower, no, it's actually quicker. The quicker the reaction will proceed to its maximum rate. Faster a Vmax will be achieved. So C is the correct answer. Number 15. Which functions are performed by glycoproteins on the surface of a cell surface membrane? To act as proteins catalyzing reactions in this membrane? Well, no, because the enzymes are present only in the inner surface, not on the outer surface. And glycoproteins don't do that. Globular proteins do that. Okay. To have a specific site where chemicals can bind, yes, because these are receptors. Receptors or antigens or cell signaling molecules. Fine. Then number three, to secrete specific chemicals used for cell signaling. No, they do not secrete anything. So only two is correct, which leaves us with C as the correct option. Number 16. The diagram represents a process by which molecules can move out of a cell through a cell surface membrane. Which process does this represent? So over here we can see that there is a high concentration and then there is a low concentration, and this is carried through a transporter protein. Low concentration, and there is no ATP. So this is facilitated diffusion. Facilitated diffusion is the movement of molecules from a region of high concentration to a region of low concentration through channel proteins or carrier proteins, and these do not require any energy. So C is the correct answer. Number 17. The photomicrograph shows the appearance of onion epidermal cells after they have been soaked in solution X for an hour. What fills the space labeled X? So as you can see over here, this thing is the cell surface membrane. Cell surface membrane. Normally, this cell surface membrane pushes against the cell wall and thus takes up the content of the entire cell. But this cell over here has been plasmolized. As a result, it shrinks and leaves hollow spaces, and these hollow spaces are places where so solution X has infiltrated. So what fills the space labeled Y? It's actually solution X. So 17, C is the correct answer. Number 18, the enzyme telomerase prevents loss of telomeres after many mitotic cycles. Which cells need to transfer telomerase enzyme? Number one, cancer cells. This is correct because they have to divide a lot of times. Stem cells, this is correct as well. Then activated memory B lymphocytes. Well, after memory B lymphocytes are activated, they can divide a lot of times. So these also requires a lot of telomerase enzymes. So one, two, three, all of them are correct, which leaves us with A as the correct option. Number 19. Which processes that occur in the cell cycle are represented in this diagram? Mitosis only no, because the mass of nuclear DNA increases, so S phase must have occurred. Mitosis and cytokinesis no. DNA replication and nuclear division only no, because mass of nuclear DNA would have stayed 4, but instead it goes back to 2, which means that cytokinesis must have occurred and production of 2 cells must have occurred. So B is the correct option. Number 20. Gene codes for the production of protein P53 that binds to damaged DNA during interphase and prevents its replication. So, in a scenario in which there is no P53, damaged DNA can be replicated. Damaged DNA can be replicated. Okay. A carcinogen in cigarette smoke mutates the gene coding for the P53 protein, preventing production of the protein. Which statement explains why this mutation may cause cancer? Because cells with no P53 are unable to undergo mitosis. No, it's only that cells with no P53. No, this is incorrect totally. Then B, cells with no P53 replicate their damaged DNA and this can cause cancer. Yes, this is true. Then there's option number C, the carcinogen in P53. Wait, 1 minute. Okay, yes. The carcinogen in P50 cigarette smoke increases the rate of cell division. No, there is nothing about that. And then the mutated P53 causes uncontrolled cell division. No, this is not an oncogene. This is just a protein that prevents damaged DNA from being replicated. So B is the correct option over here for question number 20. Okay, so moving on to question number 21. Stained onion cells undergoing mitosis were observed using a microscope. Which row is correct for mitosis in plant cells? Prophase. Centrioles are visible, yes, in prophase centrioles are visible, and chromosomes pair up at the equator in metaphase, correct? 2 telomeres are visible on each chromatid, no, it's actually 4 telomeres. And then 2 nuclear envelopes form. Well, this is correct as well because nuclear envelope is made up of two nuclear membranes, yes. So A is wrong. B. Centromeres present, correct. Chromosomes chromosomes align at the equator. It's actually centromere. Oh yes, centromere is present, correct. Chromosomes align at the equator, correct. Chromosomes replicate to form chromatids. No, it's incorrect because chromosomes exist as two daughter chromatids joined together at the centromere, and then telophase, centrioles disappear. This is correct. But there is one wrong option over here, which is where the world is incorrect. Each chromosome is visible as two chromatids. This is correct. Centromeres attached to spindle fibers at the metaphase, correct. Because the spindle is fully formed, then spindle is fully formed. Anaphase, chromatids separate and migrate to opposite poles, correct? Telophase, chromosomes deconduct, yes. So overall C is correct. Then prophase, spindle fibers formed by centrioles, correct? Metaphase, centromeres attached to spindle fibers. Anaphase, chromatids are pulled apart by centrioles, correct? D, Telophase. S- Spindle fibers form new nuclear membranes. No, spindle fibers do not make new nuclear membranes. So B is incorrect, which gives us as C to be the only correct option. Question number 22. How many statements about semi-conservative replication of DNA in a eukaryotic cell are correct? Number one, the process takes place in the cytoplasm. No, it takes place in the nucleus. An adenine nucleotide will bind against uracil on the template strand. No, this stands true only for mRNA production. In mRNA, instead of a thymine, a uracil nucleotide will be lined up. So this is incorrect. Each daughter nucleus will contain half of the original DNA molecule too. If the DNA molecule contains 40% covalent molecules, each DNA molecule, daughter molecule will contain 20% covalent molecule. This is incorrect. They will also contain 40% covalent molecule because the entire DNA is replicated. So 1 is the only correct option. Number 23. A section of the polypeptide coding for the hemoglobin beta chain contains the following amino acids: pro-glu-glu. Patients with sickle cell anemia have mutated beta polypeptide chains. The section of the mutated polypeptide chain contains the following amino acids: pro-val-glu. Okay. The table shows the possible anticodons for valine. What is the corresponding DNA triplet code for the substituted amino acid in the mutated polypeptide? So, firstly, let's find the mRNA. Let's find the mRNA code. For valence CAA, it will be GUU, then GUC, then GUG, then GUA. Then let's do the template mRNA. We're reverse engineering basically. So GUU, it's actually CAA. Then CAG. Then CAC. Then instead of G, it's CAT. So these are the possible mRNA template DNA that we can get. So let's check the answers. GTT, no. GAC, no. CTC, no. And CAT, yes. This is correct. And also, you should remember this because this comes in markings as well, that sickle cell anemia base substitution in 6 amino acid. As a result, instead of CTT, CAT codon is produced, and valine instead of glutamic acid. So D is the only correct option over here. Then, which diagram correctly represents temporary hydrogen bonds during transcription? Well, firstly, let's revisit this chapter. C and G have 3 H bonds between them. AT have 2 H bonds, while AU have 2 H bonds as well. So, AU 2 H bonds, no, not 3. CG 3, correct. GC, no, not 2, it's supposed to be 3. TU, no. P and U cannot bind to each other at all. So B is the correct option. Number 25. The diagram shows part of the DNA sequence of a gene and a mutated sequence of the same gene. CG, CCG, CCG, GAT, GAT. After this, it seems that all of the amino acids are incorrect. So what will happen is that, okay, so what are the possible effects of the mutated sequence? The presence of mRNA stop codons UAG, UAA, or UGA? No, because if this were correct, no, not really. You have to find whether these stop codons were produced by finding complementary. C, A, U, A, A, C, G, C, U, C, U, 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 A, C, G, U, A, A, G, A, U, C, C. So there are no stop codons, so one is incorrect. Change in the sequence of amino acids, this is definitely correct because lots of codons are different now. A non-functional protein, this is incorrect. This is correct because after the first codons, all the codons are different, which means that all the amino acids will be different, and thus the entire polypeptide will be completely different to what is supposed to be normally produced. So C is correct as well. And then ribosomes cannot translate the mRNA. Well, this is correct. This is not correct because there is nothing talking about the fact that the mRNA is inhibiting ribosomes or something. So this is incorrect. So just 2 and 3 are correct. And for 25, D is the correct option. Number 26. The electron micrograph shows a longitudinal section through part of a plant stem. What is X? So this is definitely, let's look and identify some other things as well. This is a sieve plate. This is a sieve tube element, and this cell over here, this is a companion cell. Companion cell. So what is X? X is a companion cell. So D is the correct option. Number 27. Sodium chloride is added to a culture solution containing freshwater single-cell plant cells. What happens to the water potential of this culture solution when the sodium chloride is added, and what may happen to the plant cells after 5 minutes? Okay, culture. If sodium ion in the culture is added to the culture, then solute concentration in culture increases, and hence water potential of culture becomes more negative. Lower, thus more negative. And if it becomes more negative, then there is a change in the water potential gradient and thus a change in the equilibrium. And hence, plant cells would have more water potential in contrast in comparison to the changed culture solution, and as a result, water will move out of the plant cell into the culture solution. As a result, the single-celled plant cells will become plasmolyzed, and the water potential of the culture solution when sodium chloride is added becomes more negative. So C is the correct one. Number 28. Which statements correctly describe transport pathways in dicot plants? In the apoplast pathway, apoplast is the non-living pathway, and it happens through cell walls as well as intercellular spaces. Non-living pathway. Water may move through plasmodesmata. No, because this is telling that water moves through cells. In Apophis pathway, it doesn't move through cells. So A is wrong. Number one is wrong. In the simplest pathway, water can move through intercellular spaces. Simplest is living pathway. So this is movement of water through the living parts of the plant. So water can move from cell to cell by plasmodesmata. And intercellular space is non-living. So this is incorrect. And then the Apophis pathway may be blocked by the Casparian strip. This is correct. So number three is the only correct option. Number 29. Which statement of the movement of water from the roots to the leaves is not correct? A continuous column of water is partially maintained by the attraction of water molecules to cellulose and also lignin. Yes, this is called adhesion. Hydrogen bonding between water molecules enables mass flow. Yes, this is called cohesion. Hydrostatic pressure in xylem vessel is higher in roots than in leaves. Yes, this produces a hydrostatic gradient which moves water from the roots to the leaves. This is called root pressure. So this is correct. And then water potential changes throughout the length of the xylem vessel elements. No, it's nothing like that. So D is incorrect. This is untrue. Okay, so number 13. Which terms describe the method by which water is transported within xylem vessel elements? Co-transport, no. Cohesion tension, yes. Osmosis, no. It's actually mass flow. And it's not co-transport because co-transport only occurs in companion cells of vessels. So, number 2 is only correct, which gives us a C to be the correct option. Number 31. What is the main function of a companion cell in phloem tissue? Providing cytoplasmic contact with the sieve tube for loading. This is correct because there are plasmodesmata between the sieve tube element and the companion cell. Yes. Providing structural support. No, it's metabolic support rather. Metabolic support. Providing the nucleus for cell division in sperm. No, there is no cell division occurring in the sperm at all. Then providing the source of assimilase for storage. No, it's not the source. It just transmits from the source. So for 31, A is the correct option. Number 32. The photomicrograph shows a blood smear. Which cell is a monocyte? Well, A has a large kidney-shaped nucleus and this is larger than most cells. So this is a monocyte. B is definitely a red blood cell because red blood cells are circular and they are in much larger numbers in comparison to white blood cells. And number C, it's a neutrophil because neutrophils have a low nucleus. And number D is a lymphocyte because they have a huge nucleus that fills up most of the cell. So A is the correct option. Number 33. The table shows some information about three blood cells P, Q, and R from a mammal. Which row identifies blood vessels P, Q, and R? So P in P, the diameter is 25 millimeters, while the wall thickness is 2 millimeters. So wall thickness is pretty thick, and so this is a artery. And on the other hand, diameter for Q is 20 millimeters, while the wall thickness is just 1 millimeter. So it must be a vein. And then diameter is just 4 millimeters, but the thickness is pretty high. It's 1 millimeter. So it must be a small artery. So we can further distinguish the arteries as well. P is a large artery, and large arteries are situated closer to the heart. And then P has a higher proportion of elastic tissue, so P is elastic artery. And Q and R has a higher proportion of muscles in comparison to other substances, so it is a muscular artery. So what are elastic arteries? Elastic arteries are large arteries with a higher proportion of elastic fibers in their blood, in their walls, and it helps withstand the pressure, helps withstand blood pressure, and prevent the artery from bursting as it is close to heart. Then it evens out pulsatile flow. It smooths out the flow of blood. It smooths out the pulses. And then muscular artery. Muscular arteries are arteries that are closer to the final destination of the blood than elastic arteries, and they have a higher proportion of smooth muscles to constrict and dilate to help maintain blood flow and controls volume of blood passing. We call vasoconstriction and vasodilation from O levels. So, for P is an artery, Q is a vein, and R is an artery. So, for 33, A is the correct option. 34. Ventricular septal defect (VSD) is a heart defect that people can have from birth. People with VSD have a hole in the heart septum that separates the left and right ventricle. What could happen in a person with VSD? Blood will leak through the hole, mostly from right to left. No, it's actually left to right, because left ventricle and left atrium have a higher pressure than right atrium and right ventricles. So this is incorrect. The volume of blood circulating through the lungs will be higher in a person without VSD because left ventricle blood flows into right, right ventricle and then eventually into the pulmonary artery. So, this is correct. Less oxygen will be delivered to the body tissues because the concentration gradient of oxygen and carbon dioxide in the blood will be altered. So 2 and 3 is correct, which is D. Number 35. Which statement about the structure of the heart is correct? Semilunar valves close when the pressure in the ventricles falls lower than the pressure in the arteries. Yes, if the pressure in the ventricles is lower than the pressure in the arteries, then to prevent backflow of blood, the semilunar valves close so that blood moves in only one direction, and that is away from the heart. A muscle called the septum separates the atrium from the ventricles. It's not actually just a muscle, it's a tissue. There is a small patch of tissue in the right atrium that acts to delay the electrical impulse. No, it's actually in septum. What they mean by this is the atrioventricular node, and it's in the septum, not in right atrium. In right atrium, the other node is present. The sinoatrial node is present. The closure of valves in the veins prevents backflow of blood into the ventricles. Not at all, because there are many veins throughout the body and they might be far, far away from the ventricle. So this is incorrect. So 35, A is correct. 36. What can be found in the cell wall of an alveolus in the lungs of a human? Cartilage? No, cartilage cannot be found. Cilia cannot be found as well. Elastic fiber? Well, elastic fiber can be found. So for 36, B is the correct option. Also, you have to know that smooth muscles also not found. Then squamous epithelium found. Ciliated epithelium not found in alveoli. And then goblet cells not found. Number 37. What are the functions of cilia and the gas exchange system? One, to move mucus, yes. To trap pathogens and dust, no. This is the function of mucus. And number three, to increase the surface area. This is incorrect because what would be the purpose of increased surface area of cilia, right? So only one is correct. And for 37, A is the correct option. Number 38. Cholera and tuberculosis are infectious diseases that can spread when people have to live in overcrowded conditions. What strategies would help control the spread of cholera and TB? Covering water to prevent flies landing on it. This is just for cholera, not cholera and TB. Providing chlorinated drinking water. This is just for cholera again. This can help prevent the transmission of cholera. And then treating severe cases antibiotics. Since both the pathogens of cholera and TB are bacteria, yes, the serious disease infections can be treated with pathogens with antibiotics. So cholera and TB. This is true for controlling the spread of both cholera and TB. Just three is correct. So D is the correct number 39. Immunity to certain pathogens develops when a person is given a vaccination. Which effects of vaccination are correct? Production of antibodies to protect against future infections, yes, but over time, antibody concentration would decrease because they would be broken down. Memory cells would still remain. So that if the same pathogen is encountered ever again, that they can rapidly differentiate into plasma cells and the antibodies can be produced to destroy the pathogen without the person even having symptoms of the disease. So one is correct. Results in artificial active immunity, yes. Active immunity is the process by which someone gets active immunity by getting infected with a pathogen, which is basically vaccination. Number three, simulation of appropriate lymphocytes. Yes, this is correct because the very fact that they stimulate lymphocytes is why we get memory cells and antibodies, right? So one, two, three, all of them are correct. And finally, we have come to the final question, which is question number 40. A student wrote three statements about antibiotics. Number one, their structure depends on peptide, hydrogen, and disulfide bonds. Number two, they're protein molecules with both. This is correct because this is a protein. They are protein molecules with both tertiary and quaternary structures. Yes. Four polypeptides provide four antigen binding sites. This is incorrect because this is the structure of an antibody. There are two light chains and two heavy chains, and then two antigen binding sites are formed, not four, and they are formed from one light chain and one heavy chain. So just one and two are correct, which is B. Okay, and with that, We have finished an entire paper of 9700 AS level biology. If you do like our video, feel free to subscribe. And if you have any queries or if you would like to obtain our notes or solve QP, feel free to check out our Facebook and Instagram pages as well as our Discord server. Goodbye for now.